Hi everyone, let's get started. So today I'm delighted to welcome Dr. Parul Johri from the biology department at UNC to give our GPA seminar. Dr. Johri got her start getting her degree in mathematics at the University of Delhi, before then also getting a master's degree in biology in Mumbai. After her master's degree, she moved on to Indiana University in Bloomington, where she worked with Dr. Michael Lynch, which is a very prominent evolutionary geneticist that I'm sure many of us are aware of. She worked on paramecium genomics as a PhD student. After her PhD, Pearl moved on and did a postdoc with Jeffrey Johnson at Arizona State University. And this is where I first became aware of Dr. Johri's work. Really doing great work thinking about how we combine all these different evolutionary processes, things like mutation and drift and selection and recombination, along with the demographic history of populations, which we've typically thought in sort of theoretical population genetics, each one of those things on their own terms, really thinking about how we combine those jointly into the models that we use to model population genetic variation. And then in 2023, Pearl joined the faculty at the University of North Carolina, Chapel Hill, where she is now, and her group has been off to a fantastic start, publishing several high-profile papers already. And Pearl just got a very large, nice NIH R35 award. So congratulations for that. And then since she's a friend and colleague just from down the road, I thought it'd be great to have her here to give a GGA seminar. So today, Pearl will talk about jointly modeling the effects of evolutionary processes on genetic variation. Thank you.
Thank you so much, David. Thank you for that introduction, and thank you for being here. So I guess he kind of gave a lot of my introduction. So we're just gonna go over it again. We kind of live in the world now where we can get a lot of genomic variation data, right? So we know that we can sequence a lot of humans, and then DNA sequence variation data can really give us insights into many, many different things. For example, it can give us, can tell us about the demographic history of a natural population, how and when humans migrated or colonized new places. And that kind of information is present in the DNA, right? In other contexts, we can also use genetic variation data within a species to understand how pathogenic species evolve within their hosts. For instance, this is for SARS-CoV-2 in human populations, and you can really use population genetics to try to understand or make predictions about sort of resistance to alleles and other evolutionary dynamics, right? But to be able to do these things, right, we need to understand how evolutionary processes really shape genetic variation. And by that, I mean all of these processes that David just talked about already, which is selection, mutation, recombination, population size changes, right? So currently in population genetics, most of the time, the situation you have is that you sort of have a snapshot of a population at some given time. And the idea is to be able to predict how these evolutionary processes have acted together to shape variation to that point, right? But the issue is that all of these processes, they don't work in isolation, and they confound inference one from each other. So for instance, this is something that has been a problem for a while now, where we sort of know that we need to know the population history of a species. Sorry, let's start here, right? We sort of need to know the mutation rates in a species to really know the population sizes in those populations. And we know that we need to account for the demographic histories to be able to infer selection. We now also know that we need to account for selection to be able to infer demographic history. We know that we need to understand recombination rates to be able to accurately infer selection. And more recent studies have now shown that we need to understand the demographic history to understand recombination rates and vice versa, right? So there's this problem of sort of confounding in these and how these evolutionary processes shape genetic variation. And this ends up leading to this big problem, right? That patterns of variation can be equally explained by very different population genetic models. So for example, you could take genomic variation from a population, and that population could really very well fit a completely neutral demographic history. In this case, what you're seeing is a scenario where you would infer that population to have undergone very, very recent growth, extremely strong growth in that population. But you can also very well explain that same population or variation as a model where you have many infrequent but very, very strong sweeps. Okay, and so it's really difficult to sometimes understand which of those processes are actually dominating in the population and what explains that genetic variation. So what's the reason for this really sort of this problem, right? So we know that population history tends to affect patterns of variation across the entire genome. But it turns out that so does selection. So even though selection is acting only on some parts of the genome, which are the sites that are selected for, the effects of that selection extends across the genome. So what I'm showing you here on, starting from this side here for beneficial mutations, we sort of all know that when a beneficial mutation arises in the population, it may increase in frequency and fix in the population. And if it does that, it carries with it other sort of linked neutral variation along with it, right? And so it changes how variation looks around it. In a similar way, if you have deleterious mutations entering into the population and get sort of purged out of the population, other linked variation with that also gets purged out. And so again, both deleterious and beneficial mutations affect variation around them, basically. In this case, this sort of a behavior for deleterious mutations is called background selection. And for beneficial mutations, right, it's called selective sweeps. Okay. And now it turns out, right, that most new mutations that occur in selected regions are actually deleterious. So this is sort of like what we currently understand of most new mutations that occur in selected regions. So there's usually a decent number of sort of effectively neutral or mild to moderately deleterious mutations. There's some debate, but we think that almost about 50% of mutations are very, very strongly deleterious as well. And then it's only a fraction that are beneficial mutations that enter into the population. And so I'll be talking a little bit more about deleterious mutations just because they happen to be a lot more common. So what are the questions that I am interested in that I work on? Some questions are, what are the consequences of linked effects of selection across the genome? How can we disentangle these evolutionary processes from each other using population genomic data? And what really is the role of adaptive versus non-adaptive processes in sort of shaping genomic variation? Can we say something about that, right? So for the purpose of today's talk, I am going to talk about sort of three different, I'm going to follow this sort of layout. So first, I'm going to talk about the effects of deleterious mutations and consequences of not accounting for them in evolutionary inference. Second, I'll talk about a computational solution and its application and implications. And third, I want to talk about how to develop evolutionary null models in species that have compact genomes, like those of human pathogens. So let's talk about deleterious mutations and what they do to variation of the genome. So we know that when purifying selection, that is this action of purging of deleterious mutations, right? When that acts on the genome, that's one thing I forgot. Okay, so when it acts on the selected part of the genome, it really changes sort of the gene genealogy that is present at those sites. Okay, so here that's a selected region, and it changes the genealogy present at those sites compared to a region that is completely neutral. And when there is linkage, right, then more of these trees look like the trees that are in selected regions. Okay, so when the genealogy is shifted, right, it ends up actually resulting in more number of sort of singletons in the genome. So this is a side frequency spectrum that is the distribution of allele frequencies in the population. So what you're seeing here, the black bars show you what would happen if you have a neutral population under equilibrium. Okay, but if you have background selection, that is, you have the input of deleterious mutations in that population, right? The background, like the side frequency spectrum that you expect actually becomes left skewed. Okay, so many more singletons in that population. We know that this is similar to another scenario where, so it looks more and more like a population that has undergone an exponential growth, because that's what happens in a population of that sort too, where you move towards sort of low frequency alleles in that population. So, so we were wondering that if this is, this is your expectation now about what's going to happen to your genome, or neutral genetic variation present in that population. So that means that it might be possible that, that you would infer population growth even when there is none, right? Because that's what's happening in this population that's experiencing deleterious mutations without actually experiencing any growth, right? So we want to test some of the sort of widely used methods. And yeah, you do see a pretty strong effect in some cases. So in this plot, the black line shows you the true population history over time. And note that we're going from past to present. The arrow goes that way. And so what's happening is that the population is actually experiencing a bottleneck in this particular case. But actually, when you use your commonly used softwares, right, you will instead infer a recent growth in this population, right? So you will completely misinfer population history in this population where you're not accounting for the input of deleterious mutations and constant purifying selection acting. And of course, this is very much a function of how much of the genome is under selection. So it's not very much of a problem if, in our case, we simulate only 5 or 10% of the genome that's under selection. By the way, these simulations were done according to human-like genomes. And so we really, in reality, we're actually in this range, somewhere here, 5 to 10%. So it really doesn't matter so much in a human population. But if you do go up to about 20% of the genome being under selection, then you no longer have accurate inferences. And so we know that, okay, we did this with humans, but the truth is, coding density varies a lot across genomes, right? So again, humans, we think is definitely less than 10% of the genome that's under selection. So we're fine. But there are other genomes, other species like Arabidopsis, C. elegans, that have about 30% of their genome under selection. Drosophila yeast, like 50 to 60%. Unicellular eukaryotes are almost really 75% coding, and so probably more. And viruses, right, like SARS-CoV-2 is like 99% coding, so pretty high number of sites that are under selection. And so the question is like how exactly are these methods performing in a lot of other species that have so much pervasive selection going on in the genome. So we also wanted to test that very specifically because, the recombination rates, et cetera, are also different across these genomes. So one of the things we did was we simulated very realistic sort of genomes this time, where we really mimicked like the genomic architecture, the distribution of exons, the distribution of regulatory regions based on previous data, how much selection is really acting on a genome. And we did that for both humans and for Drosophila. We added in heterogeneity and rates of mutation and recombination across the genome. And then we added in effects of both purifying selection and positive selection based on previous estimates of whatever we know in literature. And we also included in things like masking in this case. And we really do see that in humans, while there is, again, really, like even if you add in the maximum amount of selection that you can possibly have, in human-like populations, we find that demographic history is pretty much decently inferred. So we really don't have to worry too much about human populations. But in Drosophila-like population, where like 50 to 60% of the genome is under selection, you basically will not really recover the true demographic history in that case at all. So indirect effects of selection can really bias the inference of population size changes, especially in these sort of compact genomes that have a lot of selection going on. So next, we we were thinking about, okay, so clearly like selection is important in genomes that have lots of selection. It must also be important or more important in genomes that have little recombination, right? So there are lots of organisms that don't have as much recombination, and this is just sort of some examples of those species that I could think about, which is that if you don't have that much recombination, one reason is you have a lot of high selfing rates. So those really decrease recombination. So Arabidopsis, for example, has 97% selfing. C. elegans has a very low recombination rate overall, 99% selfing. B. falciparum actually has more recombination, but does have a decent amount of selfing that goes on. And so we were just wondering what happens when you have little recombination. Now, in that sort of a context, right, there is something else that goes on beyond simply how deleterious or how purging of deleterious mutations affects nearby neutral sites. More than that, what happens is that you can start these deleterious mutations, they can segregate in the population and they can interfere with each other, okay? So there are other things like Hill-Robertson interference actually becomes a pretty dominant force in these sort of scenarios. And so what would happen in this case is that if you have multiple segregating selected mutations in the population, they will interfere with each other and they sort of reduce the efficacy of selection of each other. So even though you have a mutation with a certain stronger selection, it's going to experience much weaker selection in that population. Okay, and that's simply because if you have recombination, right, you can combine them together. If there is no recombination, you're not going to be able to combine them together, and they compete and they actually decrease each other's effect in that particular case. So what ends up, so initially people thought that this was only mostly experienced in cases where there is mild selection, but actually now we've shown, previous people have shown that it's experienced both by weakly and strongly selected alleles, and so it's quite a prevalent force, and it ends up increasing the fixation of deleterious mutations, and it can reduce the fixation of beneficial mutations. So the exact opposite of what you would expect. So we thought that, if you have these sort of organisms, maybe you're going to not be able to infer selection very well if you don't account for this linkage, right? And currently, right, all methods that sort of estimate these sorts of selection coefficients from populations, they make the assumption that all sites are sort of evolving independently of each other. Sorry. So this is sort of like how we really perform inference of fitness effects of new mutations. Right now, people use sort of a two-step approach where they use variation from synonymous sites, right? So that's like your null model. You use the synonymous sites to estimate a population history, and then you condition on that population history, and you use the sort of distribution of alleles from non-synonymous sites, and then you're able to predict what kind of fitness effects these non-synonymous variants might be experiencing. But these kinds of approaches, right, they're assuming that every site is evolving independently of each other. So it's a single locus model. So we thought that this might be affected if there's actually a lot of linkage and recombination rates are pretty low because there's interference occurring, right? And so that is what we find, which is that if we simulate sort of C. elegans like genomes, those that have pretty low, so I don't know if this will, some people will probably understand this and some won't, but like an LDDK in C. elegans takes like 3 MB, which is about the entire size of the chromosome. So they have a lot of linkage in their genomes. And so if you take that, and you perform these similar sort of inferences of selection using current methods, right? You get a pretty strong bias again. So here, the black bars show you like what the selection coefficients were truly existing or what were simulated, right? And the red and the purple bars show you what we actually estimated using these methods. So the point is that there is a bias and you don't really get the accurate selection coefficients using these methods if there is a lot of linkage. Okay, you also really end up overestimating the proportion of beneficial mutations that are fixing in that population. So this thing called alpha, it's something that a lot of people are obsessed with in the field, right? Where we talk about like what's the proportion of substitutions in this population that were because of beneficial mutations versus other neutral or deleterious mutations. And if you kind of apply this to these sort of low recombining populations, then they get pretty strongly overestimated by these methods, again, for the same reason, because Hill-Robertson effects are actually lowering the fixation probability of these mutations. So what I've shown you in this part is that the strength of selection acting on new mutations is difficult to infer in highly selfing species or in species or genomic regions with little or no recombination due to interference. And the extent of adaptation is also overestimated. So just a summary that I talked about how if you don't account for these sort of linked effects of selection against deleterious mutations, it can affect inference of both population size changes and inference and selection, right? Okay, so what kind of solution can we think about, right, if these are problems? And so we thought that, well, perhaps we can perform some sort of a joint inference where we are simultaneously inferring population history and selection coefficients while accounting for this linkage in that particular scenario or population. So we did this using an ABC method, which is a simulation-based method, because right now it would be hard to do this while accounting for linkage otherwise. So ABC is it's a simulation-based method. So like instead, if you can't write down your likelihood expressions, right, you simulate what you expect in under different parameter combinations, and then you're asking, okay, which parameter combination, parameter combination explains my current data that I have, right? Okay, so how we did this was that we simulated very specific functional regions in the Drosophila genome and in the human genome, where we took specific exons. So we simulated those specific lengths, those local recombination mutation rates for sort of arbitrary population history and selection. And so these were all the parameters that we inferred from, right? So we had a distribution of fitness effects that we were inferring and a sort of an arbitrary population size change. And the idea was that we could try to sort of understand how this, these effects of background selection, how they decay around a selected element. We could use that to try to infer both parameters of selection and population history. And the main thing is that these are all the 6 parameters that we inferred, and the x-axis is the true value and the y-axis is the estimated value. And the method seems to work pretty well, so we're able to make these kinds of inferences. We were also able to make inferences without looking at sort of this decay effect of background selection. That is, you could just take directly like selected sites and also estimate all parameters, but the amount of error definitely increased in those cases. Why is this relevant? Because if you're trying to apply these methods to viruses where you really have no idea, and most of the genome is coding or actually under selection, you have no idea what is under selection, what's not, we could, even in those cases, we could apply these methods where we have no idea what sites are neutral, which are not, right? Okay, so there were a few aspects of our framework that were sort of different from previous ones. So one advantage was that we could fit sort of multiple aspects of the data. For example, likelihood methods were only looking at the distribution of allele frequencies, but we were looking at that, and we were also able to account for LD between different sites, and also divergence from nearby species. So all of this information is going in together to inform you about these parameters. We, of course, were accounting for linked effects of selection. We didn't make many assumptions about which sites were neutral within the selected region. So which means that we can really also use this method to get the fitness effects acting at regulatory regions in the genome. Some other related advantages that we can use only exon data for inference if we want. And we can use it in non-model organisms where we don't know which sites are neutral, which ones are not. The last one is that we also made no assumptions about selection on synonymous sites. So this is especially important for sort of genomes that are more compact, right, where synonymous sites can sometimes actually be under selection. And so since we made no assumptions about that, we can even capture selective effects on synonymous sites. And so we compared our method to other methods that don't account for, that basically assume that synonymous sites are neutral. And so here, again, the black bars are the truth, the blue bars are what our method is doing, and the shaded bars are what the other method is doing, right? So the other methods will get the population size incorrect, of course, in this case, and also get the fitness effects incorrect, right? And so by being able to sort of account for this synonymous site, like selection acting on synonymous sites, we're able to get a slightly more accurate view of fitness effects of mutations in coding regions. Okay, so now that we know that the method works or should work, we went ahead and applied it to a population of D. melanogaster. And what you see here, so on the top panel is the population history. Again, saw the times going from here to there. The black line shows you what we estimated, and all of those colored lines show you what population sizes were estimated by previous methods. So you can see that previously in this population, people thought that this population has undergone recent growth. But when we use our method, we find that this population is mostly under equilibrium. So this is just to remind you that this is sort of what we would expect, right? If there was a lot of selection going on in the genome, you would expect to infer, and if you don't account for it, you expect to infer recent growth. But because we did account for it, we think that maybe that's why we're able to see a population that's more stable over time instead. So in the second panel down here is what we inferred as the selection coefficients. So again, the black bars show you what we inferred, and the colored bars show you what was inferred previously. And the main thing to see here is that we infer a pretty large number of mildly deleterious mutations in this population compared to what other people did. And that partially can be because we're including the effect of selection on synonymous sites in this particular case. Yeah, and so that's that. We also applied it to a population of humans. And there, contrastingly, we don't see anything different from what other previous people did, right? So again, it's the same, it's following the same color scheme. So you can see that previously people inferred recent growth. We also infer recent growth. Every method is inferring a different amount of growth, but I think that's because everybody's using different number of individuals also to really make that inference. And so I would say those are pretty similar to each other in terms of what they predict. And the distribution of fitness effects, this last yellow line that you see from Huber et al., they use the same exact data set as us. So we do get actually a very similar estimate to theirs. And so as we might expect, when we apply this sort of joint inference method on humans, right, we see nothing different from what people had previously done. Kind of like, what was the point of doing this? But I think it's like a proof of concept idea that you don't actually need to account for selection to get these, or these effects are not actually, the linked effects of selection are non-dominating to the point where you need to account for them actually in humans. But in Drosophila, you do. And this is just to show that the model that we actually did estimate for humans and for Drosophila, I'm showing you only one case because we have the same story in both of those cases. That the best model, right, that we end up inferring from this predicts the actual data pretty well. So this is many different statistics shown for different parts of the genome, like the functional part, the linked part, the less linked part. And the basic point is that the orange and the black distributions overlap pretty well. So if you simulate what you think is your best model, that's the distribution of statistics you get compared to what you are observing. So it's a pretty good fit. Okay. So I just want to talk about a little bit about the implications of these results and this work. So the first is that joint inference of selection and demography will be more important for organisms with high density of selected sites, right? Low rates of recombination or non-model organisms where we just don't know what is important, what is not important. But it's clear that it's not that important for species like humans. Yeah. And the second point is that it seems that there are many more mildly deleterious mutations than previously believed. I sort of forgot to point that out, but in humans too, there are a lot of mildly deleterious mutations, but that was already understood previously. And so there are some implications of that, right? So first is that okay. Sorry. So first is that when you have mildly deleterious mutations, right, they have a pretty substantial probability of fixation in the population. So they're pretty similar in terms of fixation probability to neutral mutations, right? So they can tend to increase in frequency and fix. And what's interesting is that when they do fix in a population, right, they take the same amount of time to fix in that population as another allele that would be beneficial with the same strength if it's sunny dominant. Okay, so like, if you have a 2NS of -5, okay, a mutation, it'll fix in the same time as a 2NS of +5. Okay, and so what is the implication of that is that they end up leaving these sort of similar sweep-like signatures in the genome, where you'll have a dip in diversity around that site. Right. So even if it's a mildly deleterious mutation, right, it's creating these sweep signatures. Now, these things, they don't really matter very much in like a highly recombining population, like a Drosophila population, but they will matter more when you have a low, like when you have regions of low recombination or species with low recombination, because they will extend quite a bit further out in those cases. The second implication of these mildly deleterious mutations is that they really do tend to skew the site frequency spectrum more. And it's harder to account for them because theoretically we are not able to account for mildly deleterious mutations yet very properly. And so it's harder to theoretically account for them, basically. The third is that they can lead to this thing called associative overdominance. Okay, so that is really something like this, where suppose all of these stars actually represent mildly deleterious mutations. And so what can happen is that, every time they're homozygous, they will die, right? But if they're not homozygous and they're heterozygous, they could end up, they're actually fitter as a heterozygote. And so they're sort of creating this kind of an alternative balancing selection-like scenario, where the heterozygote is favored. And so this is called associative overdominance as well with deleterious mutations. So they can sort of mimic signatures of balancing selection. The last part is that they can increase inbreeding load in the population. So what is the inbreeding load? It's basically just, it's like saying when deleterious mutations increase in frequency, what would be the load if there were inbred individuals in the population? So it's calculated as sort of like mean fitness in the population, subtracted, and you subtract that from mean fitness of inbred individuals in the population. Okay, and so that these mildly deleterious mutations can also increase sort of inbreeding load. I'm just sort of going off a little bit of a tangent here because this is just something that I've been working on recently, and just kind of telling you about that. But it turns out, right, that what you assume about what kind of assumptions we make about fitness and how we calculate fitness actually determines how much inbreeding load can exist in a population. So this is something we've been thinking about. So I think we all take this pretty much for granted, right? So we usually use this so-called multiplicative model of fitness, where we say, if there's a deleterious mutation here, there's a deleterious mutation here, like two different ones on different haplotypes. They're independent of each other, more or less. And so you multiply the fitness that you get for both of them with their dominance. And so the final fitness you get here for that particular organism will be 1 minus 2HS, right? But what we've been thinking about recently, right, is you have these, you have these old studies of gene complementation, like these gene complementation studies where people would have a null allele and try to figure out, like when people were actually figuring out what a gene was, right? So a lot of the time it was found that if you have mutations on the same haplotype in a gene, right? Okay, let me just show it to you so that it's easier for me to understand. So basically, if you actually ended up, somewhat having a harmful mutation here and a harmful mutation there, now you have 2 bad haplotypes in that genome. So you're no longer functioning the way you would normally, and so it's actually worse than what it was in the case where it's independent. So in this case, fitness is 1 minus S, and so your dominance factor is behaving differently in these kind of models. So we just wanted to see what would happen if that's what our fitness model is, and so we went ahead and did some amount of theoretical and simulation studies with this, and we find actually that just to remind you that this means that recessive deleterious mutations will sort of experience different amounts of selection. And so, right, so actually you can see from this model that one thing that would happen is that selection is acting more strongly in this case, in this gene model than in the site model. So your allele frequencies in this population will be smaller than in the model that we normally assume, right? And so that means that, the amount of genetic load or mutational load will also be smaller in this gene model than in the sites model. And that's kind of what we see. So, if you perform the simulations here, if deleterious mutations are recessive or somewhat recessive, even if they're partially recessive, right? These models basically predict a much smaller amount of inbreeding load than the normal models that we normally assume, which is independence of these sites. It's important because a lot of people have previously suggested that if you can't explain the amount of observed inbreeding load in a population using the amount of deleterious mutations you have, then it must be because of balancing selection or other reasons why you have that sort of inbreeding load, right? But if it's this model, then maybe that's not the reason. So, right. So anyway, this is a little bit off, but I just wanted to say that deleterious mutations do have that effect too, right? Okay. Yeah. I'm gonna skip this one. And then we also found, right, that like if you account for sort of deleterious mutations and haplotype like recombination rates across the genome, you actually get pretty different statistics of haplotype diversity. So I'm not sure how many of you are familiar with like H12. These are sort of haplotype diversity statistics that are used to infer selective sweeps in the population. And this is just showing you that your null distribution that you expect would be quite different in these 2 cases, where you assume complete neutrality and where you assume that there are deleterious mutations and recombination rate variation. And you see that the one that the dark red one is the one with selection going on, right? And it looks like there are a lot of outliers there, right? And what people do is they take these outliers and they say that those are selective sweeps, right? So, you do have to be careful about that null distribution that you are using to sort of call your sweeps. So, okay, after having looked at sort of all of these various sort of effects of deleterious mutations, right? We sort of propose that it's important to come up with sort of a baseline model that accounts for all of these commonly operating evolutionary processes in a population. And by that, we mean things like the genomic architecture, right? So where are the selected sites located? How much selection is acting on the genome? And then again, similar things like heterogeneity and rates of mutation, recombination, direct and indirect effects of purifying selection, population history, and other data imperfections, right? And we sort of went through in this and have sort of like, we've talked about how one can, like if you're interested in performing inference of demography and selection, what might be sort of a good way to do this. So you can use current existing theory, et cetera, to predict based on genomic architecture if there are parts of the genome that don't have any selection acting on them. And if you can find such parts of the genome, then you can use current methods on those sites that might be neutral. But if you don't find those regions, then you really have to employ some type of a joint estimator of population history and selection. And so, sort of inspired by all of this, something that my lab is now thinking about quite a bit is how do we do this in human pathogens? Suppose we want to build evolutionary baseline models in pathogens, human pathogens. It's hard, right? Because and it's relevant. So why is it relevant? Because human pathogens a lot of them have compact genomes. So there are pervasive effects of selection acting on these genomes. Many of them undergo asexual reproduction or self-fertilization. So that will reduce the effective recombination rate in these genomes. And then third, they all experience multiple drastic bottlenecks while they invade hosts, right? So it's just part of their natural process. And so I have actually worked on a few of these different viruses, and we sort of talked about how we could make these evolutionary baseline models in these cases. But this is sort of the question that I'm sort of interested in thinking about, from here on, which is how can we infer population history and adaptation in human pathogens? And so giving you one small example of what we did do, okay, I think I've spoken about both of these things. So we looked at these laboratory influenza populations where we had this data set where these influenza populations, like they were grown in a lab, right? So we knew the exact sort of population history that they experienced because somebody had counted all of the cells and things like that before. So we knew the population history. But there were many complications in this sort of in this kind of a virus model, right? So there's a non-neutrality of synonymous sites. People don't know much about it, but that's a problem. We know that there are multiple bottlenecks in this sort of a kind of a passaging sort of experimental population. We know that there were non-right pressure dynamics, and what that means is that in a lot of viruses, the variance in offspring number is very large, and that is not accounted for in these sort of traditional right pressure popgen models. Okay. So what we did is we sort of made a method that accounted for all of these things in this influenza population, but we conditioned on knowing their demographic history. So something was known, and we try to infer selection going on in this population. What I'm showing you here is just the proportion of the first two classes, like proportion of neutral mutations. So we found them to be pretty high, like almost 70% of the mutations were neutral, and about 20% were mildly deleterious. So in this case, it's okay. It makes sense because these were all lab populations. They didn't experience any, this was like under no treatment of any sort. So, it made somewhat sense in that particular case, but it looks like most of the mutations in influenza in a lab population were kind of neutral. But one wants to be able to do this in a sort of a more realistic population, not just a lab population, because these selective effects don't really make much sense in terms of like it's more in vitro than in vivo, really. And so something I'm sort of thinking about is how or can we make these sort of models for something like Plasmodium? Why is this of interest? Because, well, for one, the third point, which is that there's a lot of publicly available data for Plasmodium. We now have something like 21 or 28,000 different individuals sequenced for Plasmodium. Plasmodium is very similar, so it undergoes multiple drastic bottlenecks. So this is kind of like it's one of the things we've done here is in its life cycle, the arrows here are proportional to what kind of a bottleneck it undergoes. And so in one life cycle, it goes at least 6 or so bottlenecks, and some of them are extremely drastic, where they go from like 10 to the 13 individuals to like 10 to the 4. So the bottleneck size is huge. And we've also estimated that these genomes are reasonably compact, so 47% of the genome is under selection. And it makes sense to have an evolutionary baseline model for being able to analyze this sort of pop gen data that's coming in now for a lot of Plasmodium species. And another interesting aspect of this is that inside the human host, right, they increase in frequency a lot, right? So they undergo this sort of a growth phase. But in this phase, the main point is that they're reproducing entirely asexually. So there's no recombination inside the host. And that's kind of where resistance to drugs, et cetera, is possible, right? So it's completely under asexual reproduction. And so that sort of got us thinking about what would be genomic signatures of selective sweets that are occurring in sort of asexually reproducing populations. So this is an interesting thing, but it turns out that we've done a lot of work on recombining species, right? We've seen those sweet plots everywhere. But if you have zero recombination, we don't really have a good we don't know what an SFS or like side frequency spectrum would look like, okay? So what we did, motivated by this, is we've been able to model using a diffusion framework, this sort of a population that's completely non-recombining. Mutations are entering the population, and you have this sort of starred allele, the green part that's increasing in frequency in the population, beneficial variant increasing in frequency in population. And if we can sort of model this using a diffusion framework, and at any point of time, we can get what the side frequency spectrum of this population would look like, including post-fixation. So this is the same side frequency spectrum you've seen before, but it looks strange because it's on a log plot. But the point is that the red points are simulated data, and the green points are our theory. So we can really predict that pretty well. And one interesting thing just for theory people is that it seems that the SFS starts off being sort of very similar to a standard equilibrium SFS, but actually asymptotes towards a 1/x² power law, where x is the allele frequency. So it's a bit different from this, and we can predict where sort of this crossover would occur between these two trends in the side frequency spectrum, and how that's kind of related to the selection coefficient of the mutation, and how far up what's the allele frequency of the beneficial mutation. So the main purpose of this is that building this theory, one thing we know is that the side frequency spectrum depends very much on the strength and the frequency of the beneficial allele. So we may be able to sort of make methods that can get that information out of the side frequency spectrum in these sort of asexually reproducing populations. So quickly, implications of just this last part, that a lot of other processes like population growth and skewed offspring distribution and background selection, everything sort of left skews the side frequency spectrum. That is, it results in more singletons in the population. So unfortunately, so do selective sweeps, right? And in a non-recombining population, the skew is even more towards just the left side. Okay, so there's no skew towards the right-hand side. And so it's going to be quite difficult to really disentangle these effects in a non-recombining population. But we hope that our framework can be used to develop methods that can estimate like the timing and strength of positive selection if we know which variant is beneficial. So if you know that this particular variant is beneficial, then we can get this kind of information. And this can be applied to other scenarios like that, like scenarios with sex chromosomes, or maybe asexually reproducing human pathogens and sort of tumor populations, which is actually where, which inspired us here. Okay, so just concluding thoughts, that I've shown you that selection can really affect sites across the genome, and that it biases inference if you don't account for it. And we've tried to make a joint framework to try to account for selection and other evolutionary processes together. And we're really hoping that we can try to do that for human pathogens and make sort of an evolutionary baseline model for hopefully Plasmodium. So that's the plan. Thank you so much. I want to quickly give the acknowledgements to all the people whose work I presented today. So those are all my students, over on your left-hand side. These are all my, this is my postdoc mentor and my current collaborators, Kavita and Brian. And I want to say that via hiring both postdocs and graduate students, please contact me if you're interested in doing anything like this. Please contact me. Okay, thank you.